Demonstration of TrenaViz, a web-based application of the underlying transcriptional regulatory network constructing software TRENA. To start, this is available at trena.systemsbiology.net, and the version I'm going to show you is a work in progress done by Paul Shannon. To start, this is the screen where you begin to identify what and choose what gene you want to model. To start with, we have a few pre-selected genes. These are from the IGAP Alzheimer's associated genes, and we are going to choose TREM2 as an example. So if we choose TREM2, then in IGV it goes to the location of TREM2 in the genome. The next thing we're going to do is we need to define the regulatory regions. And in this case, we are going to define those regulatory regions by including the enhancers as defined by gene enhancers. Now, in order to see, so we've zoomed out, and you can see TREM2 right here, and it's zoomed out to a window. We don't quite see what the rationale is behind that, but we will then add this track here that says enhancers, and again, this is from the gene enhancer. As you can see here, these are regions of the genome enhancer regions, distal and proximal, that have been identified through 3C to associate with TREM2. And for illustrative purposes, we're going to make this track a little smaller. Go down to 100. There. Now we're going to add some other tracks. We're going to add the DNase hypersensitivity regions as identified by ENCODE. We're going to add another track, which are the IGAP Alzheimer's associated variants, and you can see those here. And now we are ready to create a model. So here we're going to make a few selections for how we want to create a model of TREM2, a transcriptional model of TREM2. We're going to start by choosing footprints. So these are those, again, identified by ENCODE, that we reprocess them using two different methods, either Wellington or Hint, and with two different seed sizes to maximize the number of putative footprints that are found. For this model, I'm going to choose Hint 16 and 20, and then we're going to choose a data set. In this case, we're going to choose the temporal cortex region of the brain. This is a University of Florida Mayo RNA-seq data set, postmortem brains, and there's about 264 samples in there. And next, these footprints that we chose, we're now going to choose to only consider those footprints that are in an enhancer region. And then while those footprints show potential areas where transcription factors could bind, those transcription factors are not identified unless we link them with motifs. And here we have two options of linking those motifs. One is MotifDB, which is a more standard conservative mapping, and then TF class, which is a more liberal. And in this case, we're going to actually choose both of the above, and we're going to name our model TREM2, all enhancer. Now we're going to build the model, and you're going to see some code here that is the actual code that is running behind the scenes, and this is going to take a minute or so. If you scroll down, it continues to output what it's doing. And as I said, this will take just a minute or two. And once it's done, there it is. Now we have a model that is found here on the right-hand side. And this has identified all the potential transcription factors. And what we're going to do is we're going to toggle the IGV table so we can see the full table of the transcription factors that were chosen. And as you can see here, these are the transcription factors, and they are ordered, I believe, to start with, they are ordered by their Spearman or their Pearson, one of the two, but we can order these by any which way we want. Now, Trena is going to output all of these different feature selectors. We have beta lasso, that's the beta value for doing lasso regression. We also have a lasso p-value, which is a different package, Pearson and Spearman coefficient, and then the random forest score, as well as a beta ridge regression. The other thing we've added for convenience is the number of binding sites considered within the enhancer regions. So I'm going to organize this based on the random forest score, and that makes IKZF1 the top transcription factor by the random forest score. I'm going to now go back to the model here, and we're going to scroll down just a bit so we can see this and show some other features that we have in here. So now we're going to visualize all the footprints for top 4 transcription factors. So if you choose footprints down there and you click on IKZF1, you now have a track that shows the footprints for IKZF1. And you'll notice that some of these are, whether they're in an enhancer or not, they are just everything within the window. Do that for IRF5, LIL1, and SPI1. The other feature that TrenaViz has is we can also pull in ChIP-seq data. And that ChIP-seq data is from the ENCODE database. Now, they're not necessarily in the same tissue type or cell type that we're modeling here in the temporal cortex, as TREM2 is primarily expressed in the brain and microglial cells. But we are going to try and pull in ChIP-seq data from a couple of different cell types that are found in the database. So I'm just clicking all those individually, and they will eventually show up. But it takes some time because we're actually doing a full query. So you can see here, within the database, we found ChIP-seq hits for IKZF1, LIL1, and SPI1, but we did not find them for IRF5 because they're not found in our database. So once you have these up, you can then also mouse over these and get some additional data on them. So this IKZF1 was found in K562 cells, and it's at this location. And you click on any one of the hits, the regions, to see, give more detail. You can do the same thing with the footprints. Again, they see the location, and in this case, this is the motif that it was associated with. So now that we have our TREM2 model, what's kind of interesting is you can see a lot of these ChIP-seq hits line up with one of the enhancers that are found for TREM2, same here. But what's also interesting is, notice there's no enhancer region here, but there's definitely open chromatin, and we see both ChIP-seq and footprints in this region. The other thing we can do is we can zoom in to more specific ones, or we can zoom out, and so we're going to choose a region here to really focus on. And oops, let me zoom out there. And you can see this is the enhancer region where you have a number of SPI1 and LIL1 and IKZF1 ChIP-seq hits. You have open chromatin as well as the 3C data. There's also a few GWAS hits that are sort of in this area. So this should give you an idea of some of the tools we can use that we've built together to bring together all these data types to form hypotheses around various regulatory regions and hopefully better understand gene regulation and transcription factor binding and how that can impact a disease like Alzheimer's.